these guys won the Nobel Prize last week. The Nobel Prize for medicine, I think it was. Oh, sorry, physiology or medicine. Is it physiology or medicine? I thought it was and medicine. Anyway, it says along the bottom, Victor Ambros and Gary Ruvken, both Americans, I think, discovered this stuff called microRNA back in about, well, they developed it in about the year 2000 and onwards. It was actually discovered significantly about 5-10 years earlier than that. And this is hugely exciting to me because I'd never heard of this at this point. So I then, once I saw this, I thought, well, if they've won the Nobel Prize, microRNA to me was a laboratory experiment in the 1990s, which dozens of labs tried, and they weren't getting consistent results. So we called it antisense RNA back then, which wasn't exactly the same experiment, but that's what I linked it to in my head. And I thought it had died a death because we weren't getting any good results out of it. However, these people won a Nobel Prize for this, so something must have happened that I'm been completely unaware of. And indeed, it is something that's happened that I've been completely unaware of. It's microRNA is principally something, just to introduce the principle, is another way to silence genes, or to wake them up again, for that matter. And gene silencing is a way of shutting off quite large numbers of genes in our genome that we don't want to use again. There are about 20 to 25,000 genes in our genome, in every one of our cells. And those are the genes that contain all the information we need to build a new human, and how we were built. However, you only need some of them during development. You don't need the same genes that we have when we're fully grown than you do when you're developing in your mother's womb. They're totally different sets of genes, different purposes, and different types of genes. Something turns them off and something turns them on again. Now, oh, I quite like this. So we do know, because there are problems in the development in utero, that there is a mechanism, and doesn't always work properly. When it doesn't work properly, inevitably the developing fetus fails. It's probably what a lot of miscarriages are about. Something's going wrong. So we know it is involved in development, and it was originally discovered in an experimental a worm model that is used widely by experimenters quite a lot in this country. I think we more or less invented it, which was it Brenner in Cambridge who's got the sinusitis, Sydney Brenner, I think, and in Cambridge sequenced an entire worm. And back in the days when sequencing an entire organism was quite something, and this worm only has a small number of cells, so we've been able to follow gene expression in this very simple environment using this worm for probably 30 years now, maybe longer than that.
It was a nightmare for the technicians because they had to follow each cell under the microscope and literally just sit there and follow where the cell went. It was really arduous.
30 cells, something tiny like that, or as I remember that completely wrongly. Probably.
It's more than 30.
Probably. In addition to its usefulness in the development of a growing embryo into an adult, essentially, because it's important all the way through to adulthood. We also know that once we're grown, you can also get more problems, and those are what are behind it. We now know, as a result of understanding this area more completely, some cancers are caused by microRNA going wrong, if you like. Developmental disorders, autoimmune diseases, all these things we were saying about the immune system getting out of control in modern life, if you like, over the last 30, 40 years. A lot of people think, well, there's more asthma and there's more autoimmune diseases and so on, which may or may not be the case. But this provides an answer as to why that might be happening. And heart disease, we also know if it's involved in heart disease as well. MicroRNA is involved in almost every serious disease that you can think of, and also massively involved in the development of whole organisms. It was discovered in worms. What we got here?
They've got 1,000 somatic cells and about 1,500 enzymes.
It's not very many, then. It wasn't that far.
That's not lots.
Okay, so I can follow these slides as I wrote them. These genes need to be turned on and off correctly, or you'll get all those problems happening. For the majority of us, that works, at least for most of our lives. So this gene expression control does work very well. This is in humans. It's also vitally important in the entire animal and plant kingdom and the fungal kingdom for that matter. This stuff is fundamental to life living, if you like. So there has to be a mechanism to control expression of these genes. And what we have discovered is there are more than one mechanisms. When I was, let's go on to the next one. When I was a lad, I did my PhD. Well, I did my postdoctoral work on control of gene expression, and we picked out a particular gene that was involved in a type of leukemia, and it was overexpressed. And what it turned out that gene was a gene that turned off other genes by protein binding and all that stuff. That's an example of transcriptional control. So a gene is transcribed into a protein, and we can control that by stopping the gene being transcribed or start turning it on and off, one gene at a time, which is really useful. It's done using elements known as promoters. It can be stopped using elements known as enhancers and silencers. And the ID gene family was the one I used to work on. And they were a bunch of silencing genes. So they would bind to one of the transcription factors that bind to these promoters or stop it binding, and therefore turning the gene off. That was state of the art in its day, but that was before we discovered microRNA. MicroRNA is on a whole other level, but let's talk about something else slightly different first, because this came first in gene silencing, and oddly enough, it's another thing I worked on when I was doing a postdoctoral qualification, which is the epigenetic regulation of gene expression. That means things that don't involve DNA sequences directly. It's epigenetic. These mechanisms modify the DNA, modify molecules that interact with the DNA called histones, make the DNA hard to get at by all these promoter proteins that are trying to turn the gene on. They kind of package them away into packing cases, and the promoters can't get at them. And you can package large sequences of DNA away like this, shutting off whole parts of the genome, and that can be a long-term shutting off. So you could, there are a number of contexts that is interesting, which I'll talk about in a sec. The mechanisms that are used to carry out this silencing, one is DNA methylation, which is a modification of DNA itself, which I've mentioned multiple occasions in the last few years, and I've still never got to talk about it, I don't think, and we're not going to talk about it today either. I'm just going to mention it. Histones are these things, these packing cases, and chromatin is when you've packaged everything away and shut it away, you could call it condensed chromatin. so it's disappeared, and those genes will never be expressed again. Okay, so microRNA operates at this level. Genes are transcribed from DNA into RNA. MicroRNAs bind that RNA in a sequence-specific manner, so it's very, very specific, and stop that RNA producing proteins. So they're effectively turning them off. That's all they do. They bind sequence specifically to genes that are being expressed and stop them being turned into protein, effectively turning them off. Doesn't sound like much, but that is very far-reaching in its implications. They were discovered in the 1990s. We didn't know what they were back in the 1990s, and I think I've got a slide proving that I discovered them in the 1990s, but that's not true. But anyway, and the two Nobel laureates played a big part in working out what they were doing in the early 2000s. So this is really quite recent stuff. I did my PhD in 1987, so this has happened since I left science, essentially, which is why I don't know anything about them. That's just a picture of how DNA methylation condenses DNA and shuts it away from the promoters that I've just been talking about. How do we know? Are there any examples of this happening? And there are. There's a very famous example of genes being turned off and interfering with human health long term. One of the very tragic events of the 20th century was the World War II, obviously. But one of the terrible tragedies was the amount of starvation that was inflicted on whole populations in the world. In one in particular, happened in Holland. It was known as the Dutch Hunger Winter in 1944 to 45. In that Dutch winter, the whole population, a very large population, was starved dramatically, and such that they lost a huge amount of weight. This is referred to as severe nutritional stress. This severe nutritional stress not only affected that individual at the time, but it triggered wholesale methylation of their DNA, shutting off genes that weren't needed during a period of starvation. Anyone who was pregnant at that time gave birth to children who had low birth weight, unsurprisingly. Studies of people who were in utero during that hunger winter show that some of their genes were heavily methylated, heavily shut down, which is amazing. That's a response that's not DNA specific, that the environment has had on the heritable characteristics of human individuals. More amazingly, those changes stayed with them over the next generation or two. So the effects of the famine exposure during pregnancy extended to the next generation. The children of those exposed to famine showed tendency towards similar problems in the next generation when they had children. This suggested it wasn't a DNA-specific problem. It was a long-term problem, and it was referred to as epigenetics, above genetics. Says it all well and good. It's just me getting excited writing slides. What's so exciting about microRNA? Well, there are hundreds of different types of microRNA that have now been found. They are only expressed in particular cell types and tissues. So there'll be microRNAs only expressed in your lungs and only expressed in your liver, and so on. So they're very, very cell type specific, which is interesting because that must mean that they have a very major role in the development of your lungs and your liver and every other specialized organ in your body. The other thing that we know is these things are involved in a similar way in every single animal and plant that we've looked at throughout the world. These things, when you have that sort of information, you know that these things are fundamentally important to building life forms. This is why the Nobel Prize was awarded. This is fundamental information that we didn't know more than 20 years ago. They are short pieces of RNA that interfere with expressed genes. One example of this going wrong, a particular microRNA, they're referred to as miRNA, can cause, if it's not expressed properly, it can cause chronic lymphocytic leukemia. In fact, these different mitochondrial microRNAs can work. Some of them cause cancer and some of them prevent cancer. It's as important as that. Conserved across species, every life form you can think of, these things perform more or less the same role, or seem to. So those things are vitally important. This is that little. So this is a picture of a gel, which I think is RNA. And you prepare your RNA and you stick it in the top well there, and then you apply an electric current, and it flows through this agarose gel, and then you stain it with ethidium bromide so you can see it like this. I've done this hundreds of times, and whenever I did it, and if you didn't run it off the end of the gel, you have this huge blob of tiny RNA molecules showing up in your gel. Maybe those, all those years ago in the 1980s, are the mitochondrial microRNAs that I'm talking about, but that's just speculation. It's expressed all over the place. This is human lung. I think it's human lung. The dark pigments here, this one is B, is a normal lung, shows them being expressed in the lining of airways of the bronchus. And that's just a slightly higher magnification of the previous one. So this is something that's needed to form airways. Over here, this is D. This is expression in an alveoli. That's actually in a patient who's got emphysema and lung fibrosis, and you can see there's heavy expression of microRNAs in this tissue as well. So it's linked to normal development. It's also linked to disease, and very much relevant disease to this group. Okay, let's move on a bit faster. Consequently, there's now hundreds of labs looking into developing therapies based on microRNA. If we can stop microRNA being produced that shouldn't be produced, or we can make it be produced where it's not being produced, we could cure diseases. We could do huge amounts to help our health. A single microRNA can influence up to 400 other genes. So this is where I start to struggle, because I've not really been familiar with this field until now. When I look at it, there's so much research going on now. I can't possibly express to you everything that I need to express. All I can do is say there's an amazing amount of research going on, why that research is going on, how important this microRNA is. They control probably a third of the genes in our bodies, which is amazing. All these years we've been looking at the control of one gene, when in fact there's been one family of RNA expression that's responsible for controlling their entire genome, essentially. If we can repair aberrant microRNA, we should be able to treat many disorders. But what about aspergillosis? Well, there is already work going on aspergillosis in this area that I wasn't aware of. We know that these are microRNA 155, 146A are different microRNAs. We've already discovered that these microRNAs are included in the immune system. Macrophages, dendritic cells, T cells are all highly important in the development of aspergillosis. We know these microRNAs are important in the development of those cells. We know it can influence the activity of macrophages and other immune cells, enhancing the ability to clear aspergillus and potentially reducing inflammation. So if we can work out how this is doing that, this is a treatment for aspergillosis, because this could help our immune system to destroy aspergillus in our bodies. It's as stark as that. This one helps control excessive immune responses. Well, that is the calling card of ABPA. ABPA is caused by an excessive immune response. And we know that this microRNA is fundamentally important to that process. So again, we could be looking at a potential treatment or intervention for something like ABPA. In aspergillosis, call it a host pathogen interaction, which is something else I used to study actually. Host will be us, the pathogen is the fungus. MicroRNAs are involved in how aspergillus interacts with us. So it's fairly clear that if we get a better idea of how that's happening, then we get a better idea of how we can influence that interaction and make sure that the host wins the battle. There's a huge amount of potential in this field. Biomarkers. Someone mentioned earlier on today how difficult it was to distinguish cancer from aspergillus. Well, we know that there are microRNAs which we can pick up in blood and other body fluids. They're very stable, these things. And we know that they're circulating in the presence of aspergillosis. So if we could just detect these, find which microRNAs to detect, we can actually detect in blood aspergillosis without doing invasive tests like biopsies and so on. This is a definite possibility in the next period of time. And like we've talked about already, these things are potential therapeutic targets for all kinds of different reasons that we've just gone through. They're involved in inflammation, immunity. So they are potent potential therapeutic targets. And we talked about that bit earlier on. Research and future generations. Well, it's fairly obvious. We have to know how what these things are doing, how they're doing it, what turns them on and off. And so this is the whole new layer of genetics that is directly important to all kinds of ill health for the human population. They're an exciting frontier in aspergillosis research and patient management, because we'll be able to identify aspergillosis hopefully much more easily.